Good evening, Vanshika. Okay. So I'm Rahul Saha, and today we'll be talking about the specificities of the capacitation and then followed by the acrosomal reaction and also about the female reproductive system, right? So, talking about capacitation. So, in my previous class, we had talked about how the sperm moves through the, you can say, or how the ovum also moves through the oviduct and tries to reach. Okay, it reaches the, you can say, the ampulla region, and the effect or the pathway taken by the sperm to do the same activity, okay, to do the same activity is much more, or you can say much longer compared to the pathway taken by the ovum. So we had talked about the factors like sperm motility, then uterine muscle contractions. Okay, the third factor responsible is known as spermiotaxis. Okay, spermiotaxis. In case of sperm reotaxis, what happens is the sperms also receive long distance directional cues from the flow of fluid. Okay, long distance directional cues from the flow of fluid. Okay, which happens from the oviduct to the uterus. Okay, from the oviduct or fallopian tube to the uterus. Okay, then the sperm, what they do is they display something known as rheotaxis, which is they will migrate against the direction of this flow, okay, using their specific calcium channels. Okay, they migrate against this flow. The sperms migrate against this flow by using their calcium channels, means calcium influx into the sperms. They decide the direction of their movement. Okay, so such specificity is only found in sperms, that calcium can also not only cause induction of inhibition to polyspermy, the slow block to polyspermy, but also can decide which way the sperm will travel. Next up is, you can say, capacitation. Very, very important. Now, in case of capacitation, what happens is during the, you can say, odyssey or the journey to the, or from the vagina to the ampullary region of the oviduct, the sperm matures such that it has the capacity to fertilize the egg. when the two finally meet up. Okay, so during capacitation, which happens in the female reproductive tract, so during the journey from its advent in the vagina, from the vagina to the ampullary region of the oviduct, okay, ampullary region of the oviduct, the sperm matures, okay, the sperm undergoes maturation such that it has the capacity to fertilize the egg. That is known as capacitation. You must be having the capacity, it has the capacity to fertilize the egg. Fertilize the egg. That is known as capacitation when the two meet in the ampullary region. Okay. Now, what all things happen? What are the molecular changes that will happen in capacitation? So, unlike the sperm or if you can say frogs or sea urchins, the newly ejaculated, you can say, mammalian sperm are completely immature. Okay, they are not like the sperms of sea urchin which are already mature. They are immature and they cannot fertilize the egg. Okay, means they are unable to undergo the acrosomal reactions or even sense the cues that will eventually guide them to the egg, like from deotaxis and all. So, in order to achieve this competence, this you can say capacity, or this, in order to get this capacity inculcated in them, the sperm must go a sequential physiological changes, okay, or must undergo, the sperm must undergo a sequential physiologic changes in order to be able to cause successful acrosomal reaction. Now that process is known as capacitation. Means it has acquired the competence to penetrate the ovum. Okay. Now these changes are accomplished only and only, you can say, after a sperm has been resided for some time. It has resided for some time in the female reproductive tract. Okay. Now the thing is, this process is also not 100%. Many sperms out of the 100 million null, they may not undergo capacitation, and those who cannot undergo capacitation are held up in the cumulus matrix and are not able to reach the egg because the sperm is surrounded by, you can say, sorry, the ovum is surrounded by the zona pellucida and the corona radiata, which is the, you can say, cumulus layer, okay, cumulus oophorus layer. So those sperms which are uncapacitated or non-capacitated, they cannot even penetrate the cumulus layer, let alone bind with the zona pellucida receptors like ZP123. So they are held in the cumulus layer itself. They cannot even surpass or cross the cumulus layers and reach the zona pellucida, and then they'll undergo apoptosis or cell death. Okay. So contrary to the popular belief that in the previous classes your teacher may have told you that the race is between the swift, who is the fastest. No, who can be capacitated. It's not that you are fast, you are running, but without capacitation you have no use, right? You must also adapt. That's the thing. Evolution here also plays a great part. So it's not always the race of the fastest. Okay. So one of the study, it found that nearly all human pregnancies result from sexual intercourse during a 6-day period, okay, ending on the day of ovulation. What does this mean? This means that the fertilizing sperm could have taken as long as 6 days to make the journey to the oviduct. It's not always fast. Okay, so although some human sperms reach the ampulla of the oviduct within half an hour of intercourse, that is also possible, because I told you in the last class only it reaches, but cannot happen by its own. So all this uterine contraction, this sperm rheotaxis, then this you can say sperm motility, they all play a great role in it. But the thing is, the sperm which has reached in half an hour, okay, may, or that sperm you can call the speedy sperm, or the most rapidly, you can say, flowing or running sperm, it may have little chance of fertilizing the egg, because it may not have undergone capacitation, because it only spent half an hour in the, you can say, reproductive tract. So more time you spend, more the probability of you getting capacitated. Okay, that is again a theory. Now, the molecular processes of capacitation, they prepare the sperm for the acrosome reaction and enable the sperm to become really active, or you can say hyperactive. Okay, so there are two sets of molecular changes that I'll tell you. One is the change in the lipid composition, known as lipid changes. Okay, so we talk about capacitation, lipid composition changes. That is the first change in the sperm plasma membrane when it undergoes capacitation. So the sperm cell membrane is altered, okay, by removal of cholesterol by albumin proteins in the female reproductive tract. Very, very important competitive level question. The removal of cholesterol by albumin protein. You might have heard of albumin, which is the most, you can say, abundant protein in the human blood, right? Albumin, globulin, fibrinogen, and all. These are the globular proteins. So albumin protein of the, you can say, female reproductive tract are present in the female reproductive tract. Now, what happens is the cholesterol efflux, means it is being removed, so it will be effluxed. Now, cholesterol efflux from the sperm cell membrane has been thought, it's not a real, you can say, the research is still ongoing, but the hypothesis is it is thought to change the location of the lipid rafts. Okay, you might have learnt about the lipid rafts in class 11th. So it changes the location of lipid rafts. So cholesterol efflux changes the location of the lipid rafts. Okay, of the lipid rafts. And if you are thinking what lipid rafts are, I'll revise it for you. Okay, lipid rafts are nothing but isolated regions that contain receptor proteins, okay, in the sperm that can bind to the zona pellucida and participate in the acrosomal reaction. Okay, it will change the location of the lipid rafts in the cell membrane. So lipid rafts, they contain the isolated, you can say, regions that contain receptor proteins containing receptor proteins that can bind to zona pellucida, capable of binding to zona pellucida and causing acrosomal reaction. Okay, and causing acrosomal reaction. So this is what this will be doing, the lipid raft. That is why cholesterol is efflux, so that the lipid rafts can be placed at a proper place, because the orientation is very important. In chemistry, you might have learned about, you can say, how the bonds are really formed. Okay, you might have learned about the collision theory. Then in SN1, SN2 reaction, also you might have learned about the probability of orientation, the Z factor. Okay, so if in SN1 reaction, wherever there is OH group, the bulky group, sorry, the CS3 group, methyl group, 3R groups, from that direction, if bromine comes, will the reaction happen, even if the collision is of proper energy? Suppose both the molecules have the maximum, you can say, probable and beyond threshold kinetic energy, still it won't happen, because steric hindrance will prevail and you will not be having the reaction. Same way, if the receptors for binding with zona pellucida are somewhere where else in the plasma membrane of the sperm, where the binding cannot happen properly, even if you have the receptor, you cannot initiate the acrosomal reaction, right? So that is the proper problem. Good evening, Asma. So that will be happening. Now we don't want it to happen. Okay, we don't want that to happen at any cost. We want the proper binding of the sperm receptor with the zona pellucida receptor, ZP123 in zona pellucida. ZP stands for zona pellucida receptors present on zona pellucida covering the oocyte plasma membrane. Then only acrosomal reaction will be triggered. Okay, so originally located throughout the sperm membrane. Okay, so what happens is these lipid rafts are originally located throughout the sperm plasma membrane, but what happens is, after the cholesterol efflux, what will happen? Lipid rafts will cluster over the anterior sperm head. Means, if this is the sperm head, okay, lipid rafts are everywhere, here, everywhere. Okay, now it's only one is here, rest are everywhere. So how will it bind to the zona pellucida? Only one. It won't trigger, it won't effectively trigger the acrosomal reaction. So after the event of capacitation, what would happen is, the lipid raft, okay, suppose it is like this, so most of them, okay, will concentrate on the anterior side, front side, okay, ventral side of the sperm head, so that maximum binding can happen with, you can say, zona pellucida receptors. Okay, ZP1, then ZP2, okay, ZP3, and then acrosomal reaction can proceed, because once these receptors and zona pellucida proteins, their receptor binds, then only the acrosomal reaction will be triggered. If these reactions do not happen, if this specific binding is prohibited, then you won't have any acrosomal reaction. So in order to have the successful acrosomal reaction, or the perpetuation of the acrosomal reaction, you need this at any cost. Okay, so the outer acrosomal membrane changes and comes into contact with the sperm cell membrane in a way that prepares it, okay, for acrosome reaction again. Okay, so this is the next, you can say, point that will occur due to this lipid raft changes, that the outer acrosomal membrane, it will change its composition and it will come into contact with the sperm cell membrane, so that the acrosomal reaction can be very fruitful. Next is protein changes. Okay, next is incapacitation. You have something known as protein changes. Now, what are protein changes? Now, particular proteins or carbohydrates present on the sperm surface, okay, are lost during capacitation. So there are many proteins and also carbohydrates that are present on, you can say, the sperm surface, the plasma membrane, are lost during the process of capacitation. Now, so one of the things is it is possible that these compounds, why are they lost? Okay, we just have to think evolutionarily that why are they lost. So evolutionary mechanism, I can predict that suppose these compounds, they were blocking the recognition sites for the sperm proteins to bind to the zona pellucida. So when they were blocking, they are of no use, so they need to be removed. So now they have been removed. So it has also been, you can say, suggested that the unmasking of these sites might be one of the effects of, again, cholesterol depletion. First cholesterol goes off and the proteins which are dependent on cholesterol interaction, then they also wean off. They are also thrown off. Okay. Next problem, or not the problem, next effect will be the membrane potential of the sperm cell will become more negative as the potassium ions start leaving the sperm. Okay, it becomes more and more negative. Okay, so the membrane potential, we had talked about the membrane potential in the last class. So the membrane potential of the sperm becomes more negative as the potassium ions also start to leave the cell. Why potassium ion channels? Ions start leaving the cell. So we have potassium concentration more intracellularly than extracellularly, so it can leave via its own concentration gradient, right? Now, this change in membrane potential, okay, what will it do? Why is it happening? Will allow calcium channels to be opened, okay, and calcium will start entering the sperm. Now, calcium and the bicarbonate ions are very critical towards activating the CAMP production, okay, and CAMP, the cyclic AMP, causes the fusion, membrane fusion events which are required in the acrosomal reaction. Okay, so that is what happens. So I'll tell you what can happen is you can have a cell membrane, the sperm cell membrane. Suppose this is the sperm cell membrane. Okay, so there is a channel through which you can say this is the sperm cell membrane. So this is the channel through which the efflux of sodium will happen. Okay, another channel will be there through which HCO3 influx will happen. HCO3 will come in. Okay, will come into the cell. HCO3 minus bicarbonate ion. Okay, and that will, this efflux of sodium plus will also lead to the influx of calcium plus. Okay, calcium 2 plus into the sperm cell. Now what will happen is this calcium and bicarbonate ion together, okay, what they'll do is they'll activate the, or they will activate an enzyme known as adenylyl cyclase, which converts ATP to cyclic AMP, which we have talked about during the diplotin arrest, that this causes the diplotin arrest later on. But it is important in sperm also in different way, that the cyclic AMP, okay, will do what? Will cause the event which are responsible for, you can say, acrosomal reaction, membrane fusion. Okay, so cyclic AMP is very critical because cyclic AMP will trigger the activation of specific protein kinases like protein kinase A, which will then, it's a kinase, so it will phosphorylate, it will add phosphate groups to its substrate. Okay, and one of those phosphates, okay, one of the events of phosphorylation which is very very important is there is a specific protein molecule which I had discussed with you in one of the NEET classes concerning human reproduction. Okay, that one was, you can say, once phosphorylated, okay, this protein kinase A can phosphorylate any protein, so it phosphorylates a protein known as isomo. Okay, and once isomo is phosphorylated by this protein kinase A, isomo comes, okay, it becomes, you can say, or it transferred to the, you can say, sperm head. Okay, it migrates to the sperm head and it plays a great role in sperm egg fusion. Otherwise it is not in the sperm head, it is somewhere in the cytosol after phosphorylation. Suppose this is a sperm head, here will be your isomo protein after it has been phosphorylated, and now this will play a great role in sperm egg fusion. Okay, conduction of acrosomal reaction. That is a great prospect of capacitation. That is why capacitation is so very important. Okay, when we talk about the specificities of sperm egg, you can say, interaction. Okay, so then now we have reached, okay, from where we were, we had completed oogenesis. Now we have completed the spermatogenesis also, spermiogenesis also, is spermatid 2, you can say spermatozoon. Now we have seen how the sperm has entered the female reproductive tract through the vagina, and now we have seen that how will the ovum and sperm both will migrate their way, or they'll both travel their way, they'll both find their way towards reaching the ampulla region of the oviduct. During that point, sperm rheotaxis, uterine contraction, sperm motility, and capacitation of the sperm also has happened. Now the sperm and egg are in their close vicinity. Now just imagine this fact. So now I'm giving you a sequential, you can say, concept. Now we have reached, that's why I was teaching in this sequence. Now we have reached the ampulla region, where suppose this is the ampulla region of the oviduct or fallopian tube, where the fertilization is supposed to take place. Now both the ovum and the sperm, okay, and the sperm has reached the ampulla region, but they are still far off. Okay, little bit far off, but still that is quite a distance, not a great distance, but still a distance. Now, since they have come too close, but there is a saying like too close but yet too far. So still they are not in contact. Now what will happen? What will be the proceedings that will bring them close and will make them meet so that the acrosomal reaction can proceed? Okay, now what will happen? So the next heading will be in the vicinity of the oocyte. What will happen when the sperm is in the vicinity of the oocyte? Okay, so the thing is, towards the end of capacitation, okay, the sperms become hyperactivated. That is known as hyperactivation of the sperms. Hyperactivation of the sperms. What does it mean? What do you mean by hyperactivation? Is they swim at higher velocities and generate greater force. Okay, the sperms swim at higher velocities and generate greater force. Okay, so when they do that, that is known as becoming hyperactive. Okay, that is known as becoming hyperactive. Now when you have become hyperactive, when the sperms have become hyperactive, hyperactivation appears to be mediated By again opening of a sperm specific calcium channel where? In the sperm tail now. Okay, that's why the flagellum will move at a higher velocity. So you can say opening of specific calcium 2 plus channel in the sperm tail. So now that will cause more wiggling of the turn, more sorry of the tail, that is the flagellum made up of alpha beta tubulin dimer, the microtubules. Okay. Now what will happen is the symmetric beating of the flagellum in case of hyperactivation will be changed into a rapid asynchronous beat. Okay, now it won't be beating symmetrically, you can say one way to other, it will be an asymmetric beat anywhere it is beating, right? Asymmetrical, you can say a synchronous beat with high degree of bending, it will bend a lot. The sperm, you can say flagellum, the tail will bend a lot, right? And the power of the beat and the direction of sperm head movement are thought to release their sperm or release the sperm from their binding with the oviduct, you can say epithelial cells. Okay, because otherwise some of the sperms they stay bound to the oviduct epithelial cells. So this is a concept that you might not have heard. So since this is an oviduct, okay, so the sperms are bound here after getting capacitated. They are bound. Once they are released, only then they can fuse with the ovum and go for the reaction. But if they stay bound to the oviduct epithelial cell layer, they can never cause, you can say, acrosomal reaction even when they are capacitated, because they are never going to set themselves free. So then this hyperactivation means this bending and excessive beating, okay, beating with great force or greater force will detach or help detach the sperm from the oviduct epithelial cell. Okay, so that will be the case, oviduct epithelial cell. So after this, what happens is this will this hyperactivation only causes the release of the sperm from the binding with the oviduct epithelial cells. Means only the hyperactivated sperm are seen to or are absorbed or are observed or are able to detach and continue their journey to the egg. Okay, so the thing is hyperactivation also is a great event of significance after capacitation, because if you are capacitated but not hyperactivated, you are still you can say no cause case, you are still a case in vain, right? So hyperactivation may enable sperm to respond differently to the fluid current. Now, uncapacitated sperms, in research they have been shown to move in a planar direction, okay, straight direction, allowing more time for the sperm head to attach to the oviduct epithelial cells, okay. But capacitated sperms, they rotate, okay, they rotate around their long axis, straight axis, that will enhance the detachment of the sperm from the epithelia. Okay, so this capacitation, if it is completely fruitful, then it will definitely lead to hyperactivation of the sperm. Again, a very interrelated concept. Okay, if you are non-capacitated, then you will move in this straight direction and stay attached. But if you are capacitated state, if you have reached the capacitation, okay, what happens is the sperm will be able to rotate along their long axis, means this axis only. And since they can rotate, and again after capacitation this calcium binding channel will open, So this will lead to you can say more bending plus capacitation will help it to rotate, and that will eventually lead to its, you can say, its detachment. That will eventually lead to its detachment from the epithelial cells of the oviduct. So that is the real concept here. Okay. Now, hyperactivation along with a specific enzyme known as hyaluronidase, which can break down the hyaline layer. Okay, hyaluronidase enzyme on the outside of the sperm cell membrane. Okay, that will enable the sperm to digest the extracellular matrix of the cumulus cells. Okay, now the acrosome will release the enzyme hyaluronidase. Okay, that is the main enzyme that you should tick upon when it comes to NEET examination, hyaluronidase, which will degrade the extracellular matrix, okay, of the cumulus cells, extracellular matrix of the cumulus cells. Is the cells just outside the zona pellucida covering the oocyte, okay, or the ovum of the cumulus cells. Okay. Now hyaluronidase is a specific enzyme, okay, which will digest hyaluronic acid. So hyaluronidase will degrade hyaluronic acid. Okay. So this is one of the glycosaminoglycans. Okay. Other ones are dermatan sulfate, keratin sulfate, okay, chondroitin sulfate, heparin sulfate, dermatan, keratin. Okay. Those are known as glycosaminoglycans, special sugars. Okay. Special disaccharides they are. So if you have studied, you can say ECM, you will be knowing about this. Okay, dermatan, keratin, then chondroitin sulfate. They are also present in the synovial joints to provide, okay, chondroitin sulfate, okay, and heparin sulfate. So hypersulfated heparin sulfate is nothing but heparin. Heparin is not a protein; it's a disaccharide which contains sulfur, sulfate group, and also amino group. Those are known as glycosaminoglycans. Glycosaminoglycans, and when they attach with a protein, so those are then known as plus protein. Then it is known as a proteoglycan, which are present in the extracellular matrix. Okay, so that hyaluronidase enzyme secreted by the acrosome will degrade the hyaluronic acid, the predominant glycosaminoglycan present in the ECM of the cumulus cells. Okay, so the thing is, how does the sperm know or reach the cumulus cell or the oocyte, the ovum which is surrounded by zona pellucida and cumulus cells is one of the Q is heat. Okay, there is a thermal gradient of almost 2 degrees between the isthmus of the oviduct and the warmer ampullary region. Okay, now capacitated mammalian sperm can sense thermal differences as small as, that will you will be amazed to know, okay, thermal difference. This ampulla is the region of of fertilization. So thermal difference of 2 degrees Celsius, okay, up till 2 degrees Celsius is present between the isthmus of the oviduct, uh, isthmus of the oviduct and the ampulla or the ampullary region. This is the, you can say, thermal difference or thermal gradient which is normally present. Now the capacitated mammalian sperm can sense thermal differences as small as 0.014 capacitated, you can say, mammalian sperms can sense thermal difference of 0.014, okay, 0.014 degree Celsius. So you can think of how great that is, how enthralling and amazing that is. Okay, over a millimeter. Okay, how much is the, you can say, the distance must be over 1 millimeter. They can sense 0.014 degree Celsius of thermal gradient. Such low thermal gradient, like that cannot even be detected by our thermometers, right? So, and then they tend to migrate toward the higher temperature. Now, this ability to sense temperature difference and also you can say preferentially swim from cooler to warmer sites is known as thermotaxis and is only found in capacitated sperms. To see the or just observe the benefits of capacitation, okay, is known as thermotaxis. It moves towards the warmer region, okay, because the ampullary region is warmer than the isthmus region of the fallopian tube or oviduct towards the warmer region, and that can only be achieved this milestone by the capacitated sperms. So again, hyperactivation, which will cause it to be released or detached from the epithelial cells of the oviduct, and again this thermal cue is extremely relevant, otherwise it will never be able to find or move in the correct direction towards the ovum, only present in the capacitated sperm. Okay, so not only the lipid rafts and the protein changes, but again the movement also towards the ovum, direction, you can say directional movement, is also facilitated by the capacitation process. Now, by the time the sperm are in the ampullary region, most of them have undergone the acrosome reaction. Okay. Now a second sensing mechanism known as chemotaxis may come into play. Okay. It appears that you can say the oocyte and its accompanying cumulus cells, they secrete molecules to attract capacitated sperm toward the egg during the last stages of sperm migration. Okay. So the identity of these chemotactic compounds, okay, is still under research, but one of them appears to be the hormone progesterone, because progesterone is made by the cumulus cells. Okay, so now you come to chemotaxis. Now progesterone is secreted by the cumulus cells, and that acts as a cue to attract, okay, to attract the capacitated sperms towards the ovum. Okay, towards the ovulated ovum present in the ampullary region. Now, in humans, progesterone has been shown to bind to a receptor, okay, that activates calcium channels in the cell membrane of the sperm tail. Okay, again, if it binds to the calcium channel in the sperm tail, it will lead to hyperactivation of the sperm. So it is again another way around of understanding. Okay, now the human cumulus also appears to make a substance that attract sperms, other substances also. Okay, so but still research is underway to understand what substances those are and how it can help. Okay, So the activation takes place only after the sperm's intracellular pH has increased. Okay. Again, intracellular pH will increase how? When HCO3 minus ions will start coming in again, which can be explained by, you can say, the phenomenon that capacitation is needed in order for sperm to reach and fertilize the egg. Because when HCO3 minus ions are coming in, they will increase the sperm pH, cause it to be more and more alkaline. Okay, and that will lead to phosphorylation, activation of CAMP and all. So again, then the sperm becomes completely ready, okay, to go for fertilization. Okay, so that is the thing. Now let's come to the last reaction known as the acrosomal reaction, which is the recognition of the zona pellucida by the acrosome. Okay, finally we come to the sperm has bound to the cumulus cells now. Now hyaluronidase has been released. Now what will happen when the real acrosomal reaction happens, which is the acrosome binding, acrosomal reaction, which is the binding of the acrosome and zona pellucida layer, which is the acellular layer made up of many glycoproteins, okay, covering the entire oocyte or ovum here, ovulated ovum. So what happens is, before the mammalian sperm can bind the oocyte, it must bind and penetrate, it must bind to and also penetrate the X zona pellucida. First cumulus cells, after that zona pellucida. Now the zona pellucida in mammals plays a role analogous to that of the, you can say, vitelline envelope that comes in NEET examination. So vitelline envelope is present in the invertebrates. You can note it down. It is equivalent to the vitelline envelope in the invertebrates, zona pellucida. So that is a neat question again. Okay. So the thing is, but zona pellucida, how is it different from the vitelline? You can say vitelline envelope. Again, a neat question. The answer is it is far thicker and denser structure than the vitelline envelope. Okay, thicker and denser and denser structure. Okay. Now coming to the receptors. Okay, the mouse zona pellucida that has been researched more contains three major glycoproteins that are also asked in comparative examination. ZP1, that is zona pellucida 1, that I've been talking about a lot, ZP2 and ZP3. These are the three proteins present in the, or three glycoproteins I should be saying. Okay, three glycoproteins present in the glycosylated proteins in the Golgi apparatus are glycoproteins produced by or as a result of post-translational modification by, and this glucose residues are attached to the proteins. Okay, they could be N-linked or O-linked glycosylation done by the, you can say, glucosyl transferase enzymes present in the Golgi apparatus, right? After translation, post-translation modifications, these are PTM. So the human zona pellucida has one more major glycoprotein known as ZP4. These are present in mice. Okay, humans, they have ZP1, ZP2, ZP3 and ZP4 as well. Okay, so the binding of sperm to the zona pellucida is relatively, but you can say not absolutely species specific. Okay, and one specific species may use multiple mechanisms to achieve this binding. Okay, so what happens is, like, it appears that most sperm undergo the acrosome reaction in or around the cumulus, right? Because they will release the hyaluronidase, you can say hyaluronidase enzyme to degrade the hyaluronic acid, but they probably bind to the egg. Okay, how will they bind to the egg? Through ZP2 protein on the zona pellucida. Okay, so that is very, very important protein ZP2. Okay, so in one experiment, it was found that only those mouse eggs with human ZP2 bound sperm. Okay, and if the ZP2 was removed, the binding was not proper. Okay, that was also seen. So it was found that ZP2 is the major determinant for binding with the, you can say, sperm receptors, you can say. So then ZP3 is also a great candidate but not greater than ZP2. So ZP2 is the best candidate. Okay. So in humans, there is evidence that the reaction can be induced by, you can say, various zona pellucida proteins. Okay. But the real mechanism is still complex, like the sequence, how does it happen, where does it happen. Okay, so that is still a thing of research, like how do you bind ZP1, then ZP2, then ZP3, then the real concept that ZP3 will activate ZP2 or ZP1. Okay, that is again a great thing of challenge. Okay, so in mammals, okay, it is not the tip of the sperm head that makes contact with the egg after this acrosomal reaction. Okay, this happens in cases of sea urchin. Okay, but it is the side of the sperm head. Always know, in case of mammals, it is the side of the sperm head. Okay, that makes contact with the egg. So the acrosome reaction is in addition to expelling the enzymatic contents of the acrosome, also exposes the inner acrosomal membrane to the outside. Okay, so this is how the entire acrosomal reaction pans out, and it is the side of the sperm head. Okay, always remember the side of the sperm head that makes contact with the egg, not the tip, like in case of sea urchin or other, you can say, animals. Okay, so on the sperm side of the mammalian gamete fusion process, okay, I told you there is a specific protein known as izumo, which is named after a Japanese shrine dedicated to marriage. So this protein is originally found in the membrane of the acrosomal granule. There are granules contained, it is found there. Okay, but after the acrosome reaction, so it izumo redistributes along the surface of acrosome reacted sperm, where it is found primarily in the equatorial region. Okay, equatorial region means the junction between the inner acrosomal membrane and the sperm cell membrane is called the equatorial region. Okay, and this is where the membrane fusion between sperm and egg begins. That is the equatorial region of the sperm. I'll show the equatorial region to you. Equatorial region of the sperm. Equatorial region of the sperm. So you can see here. Okay, this is the equatorial segment. Okay, this is the one which is the junction between, you can see here in the diagram, that is the junction between the inner acrosomal membrane and the sperm cell membrane. Okay, sideways you can see the sperm heads, not the tip but the sideways it binds. Okay, sideways binding with the, this side of the sperm head will bind with the egg or the ovum. Okay, so that is known as the equatorial end, and this is where the membrane fusion between the sperm and egg would take place. Okay, so here izumo will be directly, you can say, expressing itself on the, you can say, equatorial surface, and then will cause the, you can say, sperm egg fusion. Okay, so in research also it has been found that if we block izumoprotein with antibodies, the sperm egg fusion is inhibited. Okay, is kind of prohibited or prevented in humans. Okay, so the real mechanism is this izumo. Okay, it binds to an protein, it binds to a protein present in the oocyte known as Juno. Okay, and this Isono-Juno interaction, okay, this will lead to the direct fusion of the, you can say, sperm and egg. So this isomo, okay, isomo, sometimes this question comes, binds to Zuno. Zuno is present in the ooh site, okay, is present in the sperm, and then sperm egg fusion takes place after the acrosomal reaction. Okay, so now we have completed the acrosomal reaction. In the next class, we will be going towards the female reproductive tract and the menstrual cycle. Okay, I'll explain the menstrual cycle completely to you via inhibitory, or you can say negative feedback loop mechanism, okay, which you may not have learned, but this negative feedback is asked a lot in the NEET examination, which regards to the regulation of the hormone. Negative feedback of HGH, like if you have studied in class 11th, the endocrinology chapter, the last chapter, control and coordination, you must be aware of the negative feedback loop. If that is not taught, I'll teach you how to construct a negative feedback loop. That is asked a lot in NEET examination, and we'll explain conceptually the entire menstrual cycle, each phase, okay, of the menstrual cycle via negative feedback loop of hormones. So if you had any queries, you can ask me on my personal number. Okay then, thanks a lot, and let's meet in the next class.